55 means 54 is the attendance. So we listed out the organisms, the different categories which are involved in nitrogen fixation. That is biological nitrogen fixation, isn't it, Nana? So under free living bacteria, Nana, free living nitrogen fixing. Under bacteria, just check the list of yesterday you wrote. One we wrote was, tell me, Nana, azotobacter. That was aerobic, right? Aerobic, free living, we wrote one, azotobacter. What was second example, Nana? Check the list once, all of you, Nana. Bajerincia, isn't it? Then second category we wrote aerobic photosynthetic. Tell me, Nana, what examples I gave you there? 1 chlorobium. Then 2 we gave was rhodospirillum, right? And 3 I gave you was rhodopseudomonas. Okay, Nana? Here the thing is, rhodospirillum is strictly an anaerobe, Nana. That is the thing. Better you write it this way. Anaerobic photosynthetic. And once photosynthetic, of course, here, Nana, be careful because this point specifically emphasized in NCERT textbook. So we mixed up this along with aerobic, but that is anaerobic, free living nitrogen fixing. Then could be rhodopseudomonas along with chlorobium, right? There is a little difference of those two, Nana. Can you make out from nutrition in bacteria last year? 4 modes of nutrition were seen in bacteria. In fact, Monera is the only group in which you see all 4 kinds of modes of nutrition. Photoautotrophic, chlorobium, chromatium, if you remembered.
Yes, sir.
Photoheterotrophic, rhodospirillum, rhodopseudomonas. So that's how the order has gone, Nana. So that's why better is right here in the brackets. Photoautotroph. Whereas these two are photoheterotrophs. Because it will be useful from that chapter point of view also. This is also. Rhodospirillum, Rhodopseudomonas are photoheterotrophs, Nana. Could you make out the difference, Nana, from what you learned last year? What is difference of photoauto and photohetero? Yeah, a call coming. Let it go, Nana. One second. Okay, Nana?
Yes.
So make a note of this, Nana. Is Dikshita there?
Yes, sir.
This is what better. It is a strictly anaerobic.
Yes, sir. Got it.
Did you note all of it, Nana? Because this is very, very important. Can be a question straight away. Then, of course, we were to discuss was what? Mechanism of biological nitrogen fixation as seen in root nodules of leguminous plants. So that was a different thing. So we'll now continue with yesterday's. That was mechanism of biological nitrogen fixation. As seen in leguminous plants, carried out by who, Nana? Rhizobium bacteria. From that context, if you see, we said rhizobium is free living in the soil. When it is in symbiotic association, only it will cause nitrogen fixation. And in that case, it was living as aerobe or anaerobe, Nana. When it is symbiotically associated, it is anaerobe. When it is free living in the soil, it is aerobe. Same, of course, is the case with Frankia. That point I'll tell you later. So, rhizobium is seen in root nodules of several legume plants. Like, so you can't take it as all legume plants, Nana. That is again a danger. Be careful about this. Make a note. Several legume plants. Like, list also is given. That list given in Akash is incomplete, but in NCERT, the total list we need to follow, Nana. So that's why I was telling you yesterday. Additional is there little bit in Akash, but for this aspect is concerned, better to go with Akash only. But what is missed out from NCERT, we'll be listing out here. So from that context, if it is like 1 alpha alpha, no need to bother about scientific names, Nana. The unnecessary waste of time for you about those scientific names, they are not important. Alpha alpha actually. Two. Sweet pea. Remember what was scientific name of sweet pea? Lathyrus odoratus. That you are going to come across in genetics chapter, Nana. So better for this you write. In which linkage was discovered. Lathyrus odoratus. Linkage discovered. Studied. First time. So that's why here scientific name important. Garden pea. This of course you know it very well. Physum sativum. You are aware of it. Needed also. Again in genetics you come across Mendel's all hybridization experiments work mainly in Physum. Mainly in Physum. We conduct in other plants also. So in Akash it is simply given as peas. Which peas? Not mentioned. Sweet pea, garden pea. Any more peas are there, Nana? Are you aware of? Wild pea also there. Lathyrus sativus, which we call it. We don't need to bother about that here. Okay. Then two kinds of beans. Broad bean. Clover bean. In addition, sweet clover and lentil. Lentil you are going to come across. So better is write the scientific name in genetics again. Lens culinaris. This is with codominance. In genetics you are going to come across. With codominance. So for that scientific name necessary. So altogether how many examples are there, Nana? 7 examples. You need to memorize them. Several legume plants. Make a note. As it is, Lens culinaris, the scientific name. So scientific names are important for sweet pea, then for lentil. తెలుగులో మనము ఎర్రపప్పు అంటాం లాల్ దాల్ తెలుసా ఎవరికన్నా? It is very common in North and South American countries. Several legume plants like this. Several. In fact, if you remembered, what were examples of root nodules in first year, Nana? Arachis, hypogaea, groundnut. So better you add that also. It can be from that link based, it can become. So eighth example, peanut. Can be the analyze bottom line? Will you be able to read?
Yes, sir.
Peanut. Of course, the scientific name you are aware of. What was it, Nana? Nothing but groundnut. Remember, Arachis, hypogaea. So when he says several legume plants, you cannot go by all legume plants. That is the danger with this statement, Nana. Noted this, all of you know? Rhodospirillum should be taken as anaerobic. So I think I can rub off this. Yes, no. So this list also important to remember. So overall mechanism of biological nitrogen fixation, which is involving the enzyme nitrogenase, if you take. What are the three components required? We were talking yesterday, that was the last point we were discussing. For biological nitrogen fixation, the three components required were, tell me, Nana, a strong reducing agent. In which form was it? Last point of yesterday.
NADPH.
Good. Then energy in the form of ATPs. That comes, of course, from aerobic respiration of the host as well as rhizobium, but it is a rhizobium can't be aerobic then. So it is from host. What else was required, Nana? Enzyme nitrogenase. Now, enzyme nitrogenase is with which metal ion cofactor, Nana? Gurtunda, molybdenum, activator of nitrogenase and nitrate reductase. We were saying under mineral nutrition when we were talking about molybdenum. So, is with molybdenum and iron. Molybdenum, iron. as cofactors. So molybdenum iron containing is nitrogenase. So iron also, of course, as it is iron, we were talking about all those which help in electron transport are associated with iron, and iron is reversibly oxidized. How it is, we have seen already. So enzyme nitrogenase is with iron molybdenum as metal ion cofactor. The overall reaction of nitrogen fixation, if you take by nitrogenase is N2, the so-called molecular nitrogen. What is the specialty of triple bonds, Nana, between two nitrogen atoms? Considered as what? Strongest covalent bond. That triple bond considered strongest covalent bond. That is the reason why any organism cannot use molecular nitrogen. Only those with nitrogenase can use molecular nitrogen, because that only is capable of breaking the triple bonds. And of course, which is requiring energy input. So this requires 16 molecules of ATP. Of course, 8 NADPH, which is not mentioned for us in the equation, so we don't consider that. Then during this time, it is added with 8 electrons, 8 protons, literally 8 hydrogens are added that are from NADPH, Nana, to give rise to 2 molecules of ammonia, then hydrogen, then 16 ADPs. Of course, 16 IP. What does IP mean, Nana? Inorganic phosphate.
Yes, sir.
So ATP is literally split into ADP, inorganic phosphate. So if it is this question, you tell me, Nana, in order to get 1 molecule of NH3, how many ATPs you need to supply?
16, 8.
Is it 16 for 1 NH3?
8, sir. 8, sir.
That's how. That statement also is in the textbook, Nana, NCERT textbook.
Yes, sir.
So each NH3 formation, molecule formation, requires energy input of 8 molecules of ATP. Naturally. Okay. And we know already nitrogenase is sensitive to presence of oxygen. That's why rhizobium, if it has to carry out the nitrogen fixation by its nitrogenase, it should be able to live as anaerobe. Moreover, it should be prevented from oxygen reaching the organism. If oxygen reaches the organism, what's going to happen, Nana? Its nitrogenase doesn't work. Then it will be living as aerobe. When it is living as aerobe, it can't fix atmospheric nitrogen. That's why when it was in the soil living freely, it was not able to carry out nitrogen fixation. So who was acting as scavenger, Nana?
Like hemoglobin.
Scavenger yesterday we were talking about.
Like
Tell me, what was
hemoglobin.
that?
Nitrogen.
Scavenger.
Like hemoglobin, sir.
Like hemoglobin. It is like hemoglobin-like occurring in legume plant, therefore it is called leg hemoglobin. Heme part of it is from Rhizobium bacterium. Globin part is from the host cell synthesized. That point we discussed yesterday, right?
Yes, sir.
So this is happening in presence of nitrogenase. Now we see how the mechanism is going on. Stepwise, we are going to see with diagrams as it is from textbook. Stepwise manner, the events involved in this. Enzyme nitrogenase. With the sites for N2 to be taken into this, then only it will be converted into. In the step 1, simply N2 is taken. So N2 fits into the reaction sites. So this is one nitrogen, another nitrogen with triple bonds as it is there. So nitrogen is first taken. So nitrogen gone into the reaction site. It has affinity to that, otherwise it can't fit into that. Now, 2 electrons, 2 protons, nothing but H2 added. This can otherwise be called 2 hydrogens, Nana. Should not get confused or added now. See how it's going to be now. One bond is broken. Two hydrogens are added, one each to a nitrogen. Right, like this. So we have added 2 electrons, 2 protons. Literally 2 hydrogens are added. One each is linked to a nitrogen atom. So 2 bonds out of 3. Now you are left with 2 bonds. This is called diamide. So this stage it should be taken as 2NH diamide. It is. Third step, 2 more hydrogens. So 2 more electrons, 2 more protons, nothing but 2 hydrogens more. What are you likely to get, Nana, now? 2NH2. Which can otherwise be shown like this here. This is called hydrazine. Each with 2 hydrogens. Hydrazine, you got. In the final step, 4 electrons and 4 protons, literally 4 hydrogens added. Total 8. They were from NADPH now. You get 2 molecules of ammonia now. Plus H2. So each nitrogen atom is with 3 hydrogens. They are still there in the reaction site. With the supply of energy, they will be released enzyme back to its original structure. So it is this back to its original structure. So this is removed now. Plus 2 hydrogens also. Yeah, right.
Yes, sir, towards your right.
Okay, no? Is it okay? Oh, sorry.
Yes, sir.
Okay, one. So here, source of energy. That is ATPs is respiration of host. Number one. Source of hydrogens, that is electrons and protons, is NADPH plus H plus. Eight molecules of NADPH plus H plus are used here. One in step one, no, sorry, two here, two there, and there also. Two plus 4. Here some interpret it as 6 electrons, 6 protons, Nana. 2 plus 2, in which case no hydrogens you are going to get. But actually happening is this only. So you are getting diamide, hydrazine, and finally 2 ammonia molecules from 1 and 2. Triple bonds broken.
Yes, sir.
Source of energy ATPs is respiration of the host. Source of hydrogens is NADPH plus H plus. And of course, nitrogenase enzyme was there in the bacteria. And same is also with regard to Frankia. Where was Frankia, Nana? Of non-leguminous plants. What were non-leguminous plants with Frankia in the root nodules? Elnas, Myrica, Casuarina. A non-leguminous plant with same rhizobium also was there in the root nodule. What was it? Remember? Parasponia.
Yes, sir.
Non-leguminous root nodule rhizobium. Parasponia. Non-leguminous root nodule Frankia. Ulnus, Myrica, Casuarina. Ulnus, important to remember, it is there as it is in NCERT textbook also.
Yes, sir.
Done all of you, Nana? So now, root nodule formation. Important even from IP point of view also, Nana, root nodule formation. In legumes. Or otherwise, even for Frankie also the same. So better is right, root nodule formation. So root of leguminous plant as seen in cross section, if you see. I'll show only one root here. Cell wall beneath is cell membrane. This is the epidermis. Root epidermis. We know this is the root hair. Remember what is root hair cell technically called? Knows anybody? It is an elongated epidermal cell only called trichoblast. Kadanana? Cell which has elongated to form root hair. Is root hair uni or multicellular? Unicellular, always. Root hairs are unicellular. Shoot hairs may be uni or multicellular. Root hairs always unicellular. So now the first step is the root releases sugars, amino acids, flavonoids. These are all nutrients actually to attract bacteria. Okay, releasing these for what sake, Nana? To attract bacteria. So bacteria will accumulate around the root hair. So they are released from here only, from root hair cell region, especially from root hairs. Better draw the root hair a bit big. Cell wall, beneath is cell membrane. So Rhizobium bacteria accumulate here. From the soil. So the point is Rhizobium bacteria presence in the soil is a must, Nana. A concept-oriented question possible on this. If a legume seed is sown in sterilized soil, can it develop root nodules?
No, sir.
No, sir.
No. Sterilized soil is free of microbes. So if there is no Rhizobium bacterium in the soil, it's of no use, Nana. Even if it is a leguminous plant which is capable of forming root nodules, if Rhizobium not available in the soil, it cannot develop root nodules. So Rhizobium presence in the soil is a must, and it will be there in the soil, naturally occurring in the soil. In the soil, it was actually free living aerobic. So attracted by them, Rhizobium bacteria reach the surface of the root, especially root hair. And the host also releases lectins, which are sugar binding to the surface. Lectins of the host at this juncture. Lectins of host bind sugars to surface of root hair. Lectins, one more term, again from the host only. So sugar binds to the surface so that Rhizobium bacteria reach the surface. Now, I think I can rub off this part. No, no, no.
Yes, sir.
And all of it.
Yes, sir.
Okay. See, Nana, so many things happening in nature, naturally occurring events these are. See. So lectins are helping in sticking of sugars to the surface. So attracted by them, rhizobium will reach the surface. These are all rhizobium bacteria. On reaching the surface, the bacteria release nod factor. To cause curling of the tip. To cause curling of root hair tip. So one more term here to remember, nod factor. Already was sugars, amino acids, flavonoids, then lectins. Now nod factor, to remember the things, Nana. 1, 2 amino acids, 3 flavonoids, 4th lectins, and 5th nod factor. Out of these, which is released by bacterium? Only nod factor is released by Rhizobium bacteria. That cause curling, which is called, of course, shepherd's hook. Some call it as crook also. So what's going to happen now is the same root hair will show curling like this. Cell membrane. That curl called shepherd's hook. Some call it as crook also. I don't know why. So Rhizobium bacteria are there just beneath that accumulated all these. The importance of this book not understood, but that is the region where the bacteria will cause rupture of cell wall. They digest the cell wall. Rhizobium cells. At this stage, is that side of the board okay? Na? Rhizobium releases, secretes, already released one, not factor, releases enzymes like cellulases, pectinases. Who will be digested now, Nana? Guess and tell me. To digest wall of root hair. Root hair cell, literally. So it is nothing but breaching of the wall. So this results in breaching of wall, breakdown of wall, and Bacteria reach cell membrane. Beneath is cell membrane only we know. So that what happens now, bacteria will reach the cell membrane, means invade, breaching of the cell wall, reaching the cell membrane. See. So invading, literally it is invading the cell. See the root here now. See how. So bacteria reached the cell membrane, invaded the cell literally. These rhizobium bacteria only cause invagination of cell membrane. Cause invagination of cell membrane, infolding of cell membrane. To form a long tubular structure called infection thread. So this is induced to form infection thread. Cell membrane part. See now what is happening. Cell membrane. Cell wall already breached there. Guess and tell me, Nana, where are rhizobium bacteria now? Rhizobium bacteria have gone into infection thread.
Yes, sir.
See, they gain now entry into infection thread. So this is infection thread. Rhizobium bacteria. Tell me, Nana, who induced curling of root hair tip? Bacterium by Nod factor. Who digested the cell wall? Who breached the cell wall? Bacterium. Who induced invagination of cell membrane? Bacterium.
Bacterium.
And what was the role of host root here? It has first released sugars, amino acids, flavonoids. Among them, sugars are made to stick to the surface by lectins. Attracted by them, Rhizobium bacteria reached, and they caused first by not curling, then by enzymes, digestion of the wall, breaching of the wall, then induced invagination. Three things by bacteria. Remember, Nana, what are by bacteria? What are by host? Host released literally sugars, amino acids, flavonoids, and lectins. What about bacterium, Nana? Released Nod factor, then cell wall digesting enzymes. Nod factor, cell wall digesting enzymes are by bacteria. Sugars, amino acids, flavonoids, lectins are from host. All that are for attracting bacteria, making them to reach the root hair surface. So important is what has come from the host root, what was in turn released by bacteria. Two things bacteria released, secreted. One was Nod factor, the other was cell wall digesting enzymes, cellulases, pectinases. Then four components literally are from host. Sugars, amino acids, flavonoids, and lectins. Nod factor, digestive enzymes for cell wall by Rhizobium.
Yes, sir.
So where is Rhizobium bacterium now? In the infection thread. Rhizobium bacteria are in the infection thread. So they are taken as it is into the cortex region, Nana, by the infection thread. So this infection thread goes.
So we have another class.
What is time now, beta?
9-8.
Oh, sorry. 8 minutes gone. Okay, Nana, sorry. We continue tomorrow, Nana. Don't worry.
Yes, sir.
I'll start from here tomorrow. No need to worry. You can leave.
Thank you, sir. Bye, sir.
Thank you, sir.
Bye, Nana.
Thank you, sir. Bye.
Bye,
Thank
beta.
you.
Sorry, Monday. All the best.
Thank you, sir. Bye.
Bye. Bye, Nana.